Ever wondered what makes you, you? Well, the answer is written in this incredible microscopic instruction manual that's tucked inside almost every single cell in your body. It's your DNA. Today, we're going to decode the secrets of this amazing blueprint, and we're going to start with a puzzle that seems, well, completely impossible. So let's just jump right into it with a truly mind-bending question. It's a physical challenge that every single one of your cells has to solve right now, this very second. How on earth do you cram a huge manual into a space that's almost too small to even imagine? And believe me, I am not exaggerating the scale of this problem. If you could somehow take the DNA from just one of your cells and stretch it all the way out, it would be about 1 meter long. Yep, a full meter of genetic code inside one tiny microscopic cell. It's wild. Now, that 1 meter of DNA has to get packed into the cell's nucleus, which is only about 6 micrometers across. To give you some perspective, that's like trying to stuff 40 kilometers of superfine thread into a tennis ball. It is an absolutely staggering feat of biological engineering. So to figure out how the cell pulls this off, we've got to understand how the manual itself is written. We need to deconstruct it, break it down from its most basic letters all the way up to the complete recipes that make us who we are. So get this, the entire genetic alphabet, the language of life, is made up of just 4 letters. They're chemical bases we call nucleotides, A, G, C, and T. Every single instruction for building you, for running your body, it's all written using just these 4 letters in countless combinations. These letters pair up to form 2 chains that twist into that famous shape we all know, the double helix. Now, the chains are what we call anti-parallel, which just means they run in opposite directions, kind of like a 2-lane highway. And there's a very strict, unbreakable rule. A always, and I mean always, pairs up with T, and G always pairs up with C. These pairs are like the rungs of a ladder, holding the whole beautiful structure together. Okay, so if the nucleotides are the letters, then a gene is like a complete recipe. It's a specific chunk of DNA that has the full set of instructions for making something the cell needs, usually a protein. But this is where it gets even more complicated, and honestly, just plain brilliant. In organisms like us, we're called eukaryotes, these recipes aren't simple, straightforward instructions. Oh no, they're more like a rough draft that has to go through a really sophisticated editing process first. That's right. When a cell first reads a gene, the message it gets isn't the final version. It's more of an initial transcript, and it's got a bunch of extra stuff in it that needs to be cut out before the final, usable instructions can be sent off to the cellular factory. This whole editing process is called RNA splicing. So first, the cell makes a copy of the entire gene. This copy has the parts you need, those are called exons, and the parts you don't, those are called introns. Then, the cell acts like a film editor and precisely snips out all the introns, all the junk footage, and finally, it stitches the important parts, the exons, back together to create the final, polished set of instructions. And here's where the true genius of our genome really shines. Through a process called alternative splicing, the cell can choose to stitch those exons together in different combinations. What does that mean? It means a single gene, just one recipe, can be used to create many different versions of a protein. It's an incredibly efficient way to create a ton of biological diversity from a surprisingly small number of genes. All right, so now that we get the individual recipes, let's zoom out and look at the entire library, the whole genome. And as you're about to see, the way this library is organized holds some pretty big surprises. So genes aren't just thrown in there randomly. Some are solitary, like a single standalone book. But a lot of them are grouped into gene families. These are sets of similar genes that came about through duplication events over millions of years. You can think of them like different additions or maybe even sequels to a really successful book. Now here is a real head-scratcher. You'd probably assume that a more complex organism would have a bigger genome, right? I mean, it just makes sense. Well, it turns out that's not the case at all. It's called the C-value paradox. Get this, an onion has a genome more than 5 times larger than ours. So what on earth is going on?
The answer lies in all this stuff that scientists used to just dismissively call junk DNA. It turns out that a huge portion of the genome, up to 95% in humans, doesn't actually contain recipes for proteins. Now, we know it's not all junk, but this non-coding DNA is the reason why a bigger genome doesn't automatically mean a more complex creature.
Okay, so now that we understand what's in the manual, we can finally go back to our original puzzle. How does the cell pack that meter-long string of DNA into a nucleus you can't even see? The solution, well, it's a total masterclass in organization. The cell does this in multiple stages. First, the naked DNA strand gets wrapped around these special proteins, making a structure that looks a lot like beads on a string. Then, that string of beads gets coiled up into an even thicker fiber. That fiber gets looped and organized, and those loops get compressed even more, until you get the most tightly packed form of all, the metaphase chromosome, which is perfectly packaged up and ready for cell division. The whole amazing process kicks off with this fundamental little unit, the nucleosome. See, DNA is negatively charged, so it naturally wants to wrap itself around these proteins called histones, which are positively charged. Opposites attract. This simple pairing neutralizes the charge and allows the DNA to get packed in super tight without repelling itself. That's what creates that first beads on a string structure. And the end result of all this coiling and folding and packing is just incredible. The cell manages to shrink the length of its DNA by a factor of about 10,000. That right there is how you solve that impossible packaging problem. Now, this is so much more than just a fascinating biology lesson. Understanding how this manual is written, edited, and organized has huge, profound implications for our health, because when things go wrong with the blueprint, the consequences can be incredibly serious. Let's take the Philadelphia chromosome, for example. Here, the error isn't in the letters of the code, but in the large-scale organization of the library itself. What happens is a piece of chromosome 9 accidentally breaks off and gets swapped with a piece of chromosome 22. It's like a chunk of one book gets torn out and bound into a totally different one. This one little swap creates a faulty, broken gene, and that gene produces an overactive enzyme that leads to a type of cancer called chronic myeloid leukemia. But here's the amazing part. By understanding this one precise error in the manual, scientists were able to develop a targeted drug that blocks that exact enzyme, transforming a disease that was once a death sentence into a manageable condition with survival rates around 90%. So from a simple 4-letter alphabet to a meter-long library packed into a microscopic space, our DNA truly is life's ultimate instruction manual. And as we just saw, learning to read it can literally save lives. It really leaves you with a final, thought-provoking question. If this is what we can do now, just imagine the incredible discoveries that are waiting for us as we get even better at decoding our own blueprint.